January 23, 1960. In the Western Pacific, roughly 200 miles southwest of Guam, a US Navy support ship holds position in heavy swells. Below it, descending at less than a meter per second through total darkness, is a vessel called the Trieste. A steel sphere, barely big enough for two men to sit in, suspended beneath a float full of gasoline, dropping through water no human had ever entered. The men inside are Swiss oceanographer Jacques Piccard and US Navy Lieutenant Don Walsh. They have been falling for nearly 5 hours. They are passing through the last pressurized zone before the bottom of the world. At 30,000 feet, the plexiglass window cracks. The sound fills the sphere. They keep going. When they land, the sediment beneath them, fine as chalk dust, compressed by millions of years of weight, billows up around the viewport and doesn't settle. For 20 minutes, they sit at the deepest point on the surface of the Earth and can see almost nothing. They come back up. They tell the world they were there, and nobody fully understood what they'd found or what it meant for another half-century.
At the tone, 20 hours, 59 minutes, coordinating universal time.
The Mariana Trench is not, as it's usually described, simply a hole in the ocean floor. It is a subduction zone, a place where the Pacific Plate, one of Earth's largest tectonic plates, is being driven beneath the Mariana Plate. The trench is the scar left behind by that collision. It has been forming for roughly 180 million years, and it is still forming now. The trench runs roughly 2,550 kilometers in length. Its deepest point is a slot-shaped depression at the southern end called Challenger Deep, named for the HMS Challenger, the British research vessel whose crew first measured the trench's depth in 1875 using weighted rope. The current confirmed depth of Challenger Deep is 10,935 meters, 35,876 feet. That's nearly 7 miles of water stacked directly above the seafloor. To put that in terms that land differently, Mount Everest, the tallest point above sea level on Earth, is 8,849 meters tall. Drop Everest into the Mariana Trench, and its peak would still be more than 2 kilometers underwater. The pressure at that depth is 1,086 bar, approximately 1,072 times the atmospheric pressure at sea level. To give you a physical sense of that, the pressure exerted on the hull of a vessel at Challenger Deep is equivalent to the weight of 50 jumbo jets pressing down on every square foot of surface. For most of human history, this was assumed to be a dead zone. The logic was straightforward. No light, no photosynthesis, no food chain, temperatures hovering just above freezing, and pressure capable of crushing anything built by human hands. The deepest parts of the trench, the thinking went, were simply too hostile for complex life. Piccard and Walsh went down to find out what the trench actually contains and what it has continued to reveal in the 60 years of exploration since that first dive is a story about what life actually is and where it can go. The first thing to understand about extreme pressure biology is what pressure actually does to living tissue. At depth, hydrostatic pressure doesn't crush organisms the way it crushes a submarine. It compresses at the molecular level. Specifically, it disrupts the proteins that run every biological process. The enzymes that catalyze chemical reactions, the structures that maintain cell membranes, the machinery that reads DNA. Under sufficient pressure, those proteins stop working. Their shapes distort. The chemistry fails. This is why, for a long time, biologists believed fish couldn't survive below approximately 8,000 to 8,500 meters. Not because of the physical weight, but because their cellular machinery would simply stop functioning. There appeared to be a hard biological ceiling on depth. What researchers found in deep-sea fish was a molecule that changes that ceiling. It's called trimethylamine oxide, TMAO, and it functions as what biologists now call a piezolyte, a pressure solute. TMAO stabilizes protein structure under high pressure, acting as a kind of chemical chaperone that keeps the molecular machinery running in conditions that would otherwise shut it down. The remarkable thing about TMAO in deep-sea fish is not just its presence, it's the pattern. TMAO concentration in fish tissue increases linearly with depth. The deeper a fish lives, the more TMAO it carries. The correlation is precise enough that you can estimate a fish's maximum depth range from its TMAO levels alone. Think of it this way. Imagine the proteins in your body are precision machinery, finely calibrated, sensitive to operating conditions. Pressure is like running that machinery in a vice grip that tightens with every meter of depth. TMAO is the lubricant that keeps the gears turning despite the grip. Without it, the machinery seizes. With it, the machinery keeps running, up to a point. That point appears to be somewhere around 8,000 to 8,500 meters, where the TMAO concentration required to counteract pressure would make a fish's internal chemistry isosmotic with seawater, meaning it would lose the osmotic gradient that keeps water inside cells rather than outside them. The leading theory is that this biochemical wall, not the pressure itself, is what limits fish depth. Below 8,500 meters, in the true Hadal zone, fish disappear. But the trench doesn't empty. Something else takes over. Sunlight penetrates to roughly 200 meters in clear ocean water. Below the twilight zone, at around 1,000 meters, darkness is total and permanent. No photosynthesis occurs below that depth, which means the entire basis of the standard marine food chain, the conversion of solar energy into biological matter by phytoplankton, simply doesn't happen in most of the deep ocean. The conventional model for deep-sea nutrition is called marine snow, a continuous, slow fall of organic particles from the surface. Dead phytoplankton, fecal pellets, the remains of organisms from every zone above, drifting down through the water column like a permanent grey blizzard. Most of it is consumed long before it reaches the sea floor. What arrives at 6,000 meters is sparse, thin, a desert's worth of nutrition spread across the bottom. The Mariana Trench is a geometric exception to this. A trench is a funnel, material that falls from the surface and from the surrounding abyssal plain gets channeled by the trench walls down toward the lowest point. Organic carbon concentrations at Challenger Deep have been measured at 2 to 4 times higher than on the surrounding abyssal plain at the same depth range. The trench's shape actively concentrates nutrition at the very point where you'd expect it to be most scarce. This explains something that confused early researchers: why the deepest parts of the trench are, in terms of biological activity, surprisingly rich. Not by surface ocean standards, but compared to the open abyssal plain around them, the Hadal zones are fed. And what lives on that concentrated organic matter, at depths below where fish can go, is a community that has been assembling for millions of years in complete isolation from the world above. When the first landers with cameras descended into those zones, what they found wasn't the sparse, nearly lifeless environment the models predicted. The Hadal Ecosystem Studies expedition, led by Jeff Drazen and Patty Fryer of the University of Hawaii aboard Schmidt Ocean Institute's research vessel Falkor, conducted the first detailed biological survey of the Mariana Trench across multiple depths, from 5,000 to 10,600 meters. The results changed the picture significantly. Amphipods, small crustaceans that look something like shrimp, were found in swarms of thousands at full hadal depth, gathered around foodfalls on the trench floor. Sea cucumbers, holothurians, moved across the sediment in visible numbers. Polychaete worms were present in the deepest sediment samples. The landers recorded consistent, repeated biological activity across all target depths. At the vents, hydrothermal outlets where superheated water, rich in hydrogen sulfide and other compounds, seeps through the sea floor, something more structurally remarkable was waiting. Entire ecosystems were being powered by chemosynthesis rather than photosynthesis. The engine at a hydrothermal vent isn't phytoplankton at the surface. It's bacteria that metabolize chemical energy from the vent fluid itself. Everything else, the tube worms, the shrimp, the crabs, the fish on the slopes above, feeds on those bacteria or on organisms that feed on them. A vent ecosystem changes the old assumption about sunlight. Its food web can begin in darkness, with chemistry rising out of the seafloor. That is the part that mattered. Life did not have to start with light falling from above. This was not, for astrobiologists, an academic observation. Kevin Peter Hand is a planetary scientist and astrobiologist at NASA's Jet Propulsion Laboratory. His research focuses on the origin and distribution of life in the solar system, specifically on ocean worlds, moons and planets with subsurface liquid water, no sunlight, and potential hydrothermal activity. In 2012, Hand was aboard James Cameron's dive to the bottom of the Mariana Trench, not as an observer, but as a working scientist, studying the trench's biology and chemistry as direct analogues for extraterrestrial ocean environments. The moon he's most focused on is Europa, Jupiter's fourth largest, whose surface is a shell of water ice over a global subsurface ocean estimated to contain more liquid water than all of Earth's oceans combined. Europa receives almost no solar energy at its surface. Its ocean is in total darkness. Models suggest there may be hydrothermal activity on its seafloor, driven by tidal heating from Jupiter's gravity. The pressure at the bottom of Europa's ocean, depending on ice shell thickness, could be significant. The Mariana Trench is, as close as we can get on Earth, a physical model of that environment. What the trench demonstrated, what vent ecosystems made impossible to ignore, is that life does not have to be built around sunlight. It can be built around liquid water, chemical energy, and enough time for biology to take hold. The discovery of chemosynthetic ecosystems in the deep ocean didn't just expand the map of life on Earth, it expanded the map of where life in the solar system could plausibly exist. Saturn's moon Enceladus has an active subsurface ocean. Geysers vent water vapor and organic compounds from its south pole, material from the ocean below erupting into space. NASA's Cassini spacecraft flew through those plumes and detected hydrogen, carbon dioxide, and methane, the chemical fingerprint of hydrothermal activity. The same chemistry that feeds a vent ecosystem at Challenger Deep. The trench didn't just change ocean biology, it changed the question of whether we are alone. Go back to January 23, 1960. Piccard and Walsh sit at the bottom of the world, in a cracked vessel with sediment obscuring their view and a telephone that barely works. They spend 20 minutes there. They take temperature measurements. They attempt to measure current. They call the surface and confirm their depth. Then they ascend. 3.5 hours back up through the dark, and they tell the world they've been to Challenger Deep. Picard believed he'd seen a flatfish at the bottom. Walsh, decades later, said the sediment cloud they stirred on landing never cleared. That for all 20 minutes at depth, visibility was essentially zero. The flatfish, he suggested, might have been a misidentification. Neither of them were trained biologists. There are no photographs. What they brought back was a confirmed depth, a confirmed temperature, and a question that nobody had the tools to answer yet. What actually lives down there? It took 54 years of expeditions, remotely operated vehicles, baited landers, sediment cores, and the development of entirely new deep-sea biology to begin answering it. The Hades expedition in 2014 gave the first systematic picture. The vent discoveries gave the framework for understanding how deep-sea ecosystems are even possible. The TMAO research explained the biochemical ceiling on fish depth. Kevin Hand's work built the bridge from the trench to the outer solar system. Each piece of knowledge came slowly, from vessels that mostly couldn't see what they were measuring, from samples pulled up through 7 miles of water, from cameras that showed a few square meters of seafloor at a time. Picard and Walsh were down there for 20 minutes, in 1960, in the dark. Everything we know about the bottom of the world has been built on top of those 20 minutes. For most of the modern age, the number of people who had reached Challenger Deep could be counted on one hand. Even now, after new private and national expeditions expanded that list, the human footprint at the deepest point on Earth is tiny. Humans have landed on the Moon 6 times. That comparison still feels wrong. The deepest place in our own ocean remains one of the least familiar places humans have ever reached. Not because the technology is impossible. Victor Vescovo made repeated dives to Challenger Deep in 2019. Because the trench is still, in almost every meaningful sense, unexplored. The landers cover meters of seafloor. Challenger Deep covers roughly 11 square kilometers. The broader Hadal Zone, all the world's ocean trenches below 6,000 meters, covers an area roughly the size of Australia. What's down there, in the sediment, around the vents, in the water column above the trench floor, remains largely uncharacterized. The species count from the Mariana Trench is still incomplete. The full picture of how Hadal ecosystems are structured, how they change across depth, how the biological communities at 6,000 meters differ from those at 10,000 meters, is still being assembled. The trench has been forming for 180 million years. The communities at its bottom have been living there, under pressure, in darkness, on marine snow and vent chemistry, for timescales we are still trying to measure. Long before humans had charts, ships, or instruments that could reach them, they were already there. When Picard looked through that cracked plexiglass viewport in 1960, he may have seen something move, or he may have seen nothing but sediment. Either way, something was down there. It still is.